I'm just going to test my audio just to make sure. Okay, fine. Excuse all the dithering around. I need to just check everything before I even start, because I had my computer completely redone, and it's not really behaving the way it should. But in any case, we'll do our best. Okay, so good morning, good afternoon. We're just going to finish off our discussion about the nervous system and input and output and everything from the nervous system by talking about this one really important aspect of output, which is the induction of motor movement. We'll use as our model the skeletal muscles or voluntary muscles. But everything I say to you about muscle, about the way in which muscle movement arises, the model that we're going to use, which is called the sliding filament model, applies to all kinds of muscle movement, whether it's voluntary or involuntary. It's just that in the voluntary muscle, the action potential is sent deliberately. It is sent as a conscious action. Tell your arm to contract and it contracts, but you don't send instructions, for example, to your gut muscles to perform peristalsis. That is under a different mechanism and largely under the control of the autonomic or involuntary nervous system. Similarly for the heart generates its own muscle contractions, but the contractions are controlled, as we learned before, by aspects of the autonomic nervous system. We are going to talk about the voluntary muscle system because it forms an easy, nice story from generation of the action potential to the generation of the contraction itself in the muscle. So all muscle movement depends upon the interaction of different filaments made up of proteins, of different proteins. The fundamental filaments that we have to talk about are, first of all, these designated in yellow, which are called actin filaments. Actin actually is composed of a number of different proteins all put together, but it forms this filament here, which is often referred to as the thin filament. And you'll see the reasons for that in a minute. So that is the thin filament. The other kind of filament involved is myosin. And myosin consists of these heads here, and then a tail, which stretches behind like that. And involuntary muscle, in muscle, the myosin is actually organized to form a thick bundle of myosin molecules. So all of the tails are bound together, and it's forested with these heads sticking out. These heads are actually capable of movement. They're capable of moving back and forward, back and forward. So they go back and they get cocked, and then they are released and flip over the other way. That simple movement is the basis of the contraction of the muscle. So the myosin forms this thick filament here, and the thick filament is going to interact with the thin filament to produce movement. So we need to have a careful look at how the muscle cells are organized to understand how the movement is actually produced. So I want you to just follow along here on this diagram here for a minute. This here is actually an individual muscle fiber. It's actually a very large cell. It's a muscle cell, but it has many nuclei. It's formed from the union of, originally, of many cells joined together, organized, and lost their cross walls. So it's surrounded by cell membrane, and that is called the sarcolemma. Here's the name here. We'll refer to it again in a minute. That's the sarcolemma. That's the cell membrane of the individual muscle cell. But if we look on the inside of the muscles, of this muscle fiber, we'll see a whole lot of bundles of the different protein filaments. And these are organized into what are referred to as myofibrils, a bundle of actin and myosin filaments all surrounding one another. And you must try and think of this in 3 dimensions, not into the 2 dimensions that we squash it into for convenience sake. So this is a myofibril. Bundles of myofibrils are gathered together on the interior of the muscle cell, which is called the muscle fiber. The muscle fiber, many of them, are bound together to form thick muscle units, which all together form the muscle itself. So what we've done is we've peeled off one of those bundles of muscle fibers, but there are many of them in the muscle. They're all organized and tied to the bones at the ends with tendons. And they are obviously supplied with nerves, because it's an action potential from the nerves that is going to cause them to contract. So we're now going to go all the way down to the myofibril and see how it is organized. Here you can see there's a thin line there and a thin line there, which are called the Z lines. In here, in this micrograph of muscle, you can see them as very thin lines here in between these darker stripes. Those dark stripes are what give striated muscle its name. They are quite easily visible as dark stripes. And here they are here. This would be a stripe. This whole thing here would be a stripe. And then on either side of it, the Z lines. And that unit, Z line to Z line, with all of the internal organization here, is called a sarcomere. And the sarcomere is the unit of movement in the muscle. And if we look at the sarcomere in a more diagrammatic form, you'll see here is the Z line. The Z line is actually a proteinaceous disc to which all of the thin filaments here are attached. The thin filaments here are just visible in this electron micrograph here. And those are the actin filaments there, and they are attached. These are all attached here. These ones are all attached here. Now this here is actually the muscle as it is when it is relaxed. So there is a space here. And the striation here, there, is this whole unit here, there. So that is the sarcomere. The fiber, here's something that you must remember, okay? This myofibril here is surrounded, it's richly invested with endoplasmic reticulum, but it is highly modified endoplasmic reticulum. It's called sarcoplasmic reticulum. That means muscle reticulum. And what it is a store. It is a store of calcium, because it's calcium that actually is going to induce muscle contraction. But calcium is only released under the influence of an action potential through a complex set of steps, which we're not going to go into. But here in this diagram here, you can see, here's the sarcolemma, the cell membrane around the whole muscle fiber. On the inside here is the myofibril there, and it is surrounded by endoplasmic reticulum, sarcoplasmic reticulum. The two are interchangeable. So here's the organization of the myosin. We've got all our actin filaments attached there to the Z lines. The myosin is in between these. Now, here's what I mean when I say you need to think of it in 3 dimensions. It's not flat like that. It's cylindrical. So a myosin filament, thick myosin filament, may be surrounded by many actin filaments on the outside. So here it is in its relaxed form. What this muscle, the voluntary muscle, is under the influence of voluntary control. So there are nerves which run to that muscle, and as the nerve, the voluntary nerve, reaches the muscle, it subdivides tremendously to form a sort of a tree shape, which goes to all over the muscle. And it's the ends, the axon, here's the axon here, the axon splitting up, and it's terminating here, and the axon terminates, the axon terminal is here, a little plate, each one attaching to a muscle fiber. So these are the axon terminals here, forming plates which attach to the muscle fiber. And when an action potential is sent, instructing a muscle to contract, the action potential reaches the synapse here, but this synapse is between the nerve and the muscle. So it has to release a neurotransmitter in order to communicate across the synapse. And the neurotransmitter that it uses is called acetylcholine. And acetylcholine, when acetylcholine is released into onto the muscle fiber, it causes the sarcoplasmic reticulum to dump all of its calcium onto the myofibrils. And that is what induces the contraction. There are several steps which we're not going to discuss, but I'm just going to show you. Here, for example, is our relaxed muscle here. This is what it looks like in its relaxed form. Under the influence of calcium, which has flooded the myofibrils, the myosin pulls the actin filaments inwards. The actin filaments are attached at the Z lines here. So the distance between the Z line decreases. The Z lines are pulled towards one another as these actin filaments are pulled in by the myosin here. So the myosin is actually attaching to these filaments and pulling them this way and this way to cause the muscle to contract. Here's the contracted sarcomere, much shorter. And look what has happened. These distinct striations here have disappeared because all of the fibers now are lying next to one another. So that is the basic method of contraction of the muscle. The myosin pulls the actin filaments in from either side. The actin filaments are attached to the Z-line proteins, so the whole sarcomere contracts. And that happens over the whole length of the muscle itself. So the whole muscle ends up contracting in this fashion. So the actual mechanism whereby this takes place all relies on the action of those myosin heads that I showed you before. When calcium floods the muscle fiber, several steps arise, but the ultimate action is that ATP is used, energy from ATP is used to actually cock the head of myosin filament. So here's the myosin filament in its relaxed, low energy configuration. That's how it is when it is unattached to anything and not doing any work. But when ATP is used, that head can be moved back, and it is now like a spring loaded in the open position. So once that happens, the ATP is used, but once it reaches this conformation, it is actually able to bind to actin. And here it is here. It's binding to the actin. And once it binds to the actin, it actually releases the ADP completely. And without the attachment of that ADP, the head resumes its normal position. It flips over, but remember it's attached to the actin, and it moves the actin in this direction here. So once that happens, then it actually releases from the actin. And as soon as it releases from the actin, as long as the instruction, as long as action potentials are still arriving at the axon terminal on the muscle fiber, as long as action potentials are still arising, then it will again bind ATP, flip back, and move the actin again. As soon as that as soon as the action potentials stop, the sarcoplasmic reticulum pumps out the calcium very, very rapidly. So the calcium is removed from the cell, secreted into the sarcoplasmic reticulum, and can no longer spark the generation of contraction. So now what we need to realize, okay, we're going to, here's muscle relaxed and long. Contract the muscle like this, and as long as the action potentials are still going, because your brain is instructing your muscle, contract, contract, contract. As soon as you stop that, the muscle will relax. All of the binding here will stop. But the muscle doesn't go back to its normal shape. There is no power in the myosin to push the actin filaments apart and lengthen the muscle. Instead, in order to lengthen that muscle into its relaxed full length, we have to contract muscles on the other side of the arm. And those actually physically pull the muscle filaments back into their open position like this, ready for another instruction to contract. So there is some complexity to it, but it's actually a very logical, simple mechanism if you go through each of these steps. So there is a little movie here for you. I hope that this works. Let's just see. Okay, so that is that to terminate our discussion of the nervous system. Now, you're not getting off the hook entirely because this is very short, but you can view this just as an independent video. But I am going to put up another class, which is at the start of our discussion about plants. So please make sure that you view that video before we meet on Wednesday. Okay, I'll try and do it this evening. Today is Monday. I'll try and do it this evening. I may have to do it tomorrow morning on Tuesday, but please make sure that you watch both of these videos before we meet on Wednesday. Okay.